All right, so welcome everyone to what I believe is the first genomic seminar series of 2022, which is brought to you by the Center for Genomic Science Innovation, or CGSI. So hopefully we'll be back in person before too long, but in the meantime, I think we have a fantastic virtual speaker lined up for today. So it's my great pleasure to introduce Hilary Finucane, who is an associate member of the Broad Institute, an assistant in investigation at Massachusetts General Hospital, and a member of the faculty of Harvard Medical School. So Hilary's research focus is on the development and application of statistical and computational methods for understanding the genetics and mechanistic basis for common diseases and complex traits. And I think in particular, she's become well known for her recent work in integrating functional genomic data, such as gene expression or epigenetic data, with GWAS data and other large-scale genetic data sets for identifying causal variants and relevant cell types for diseases and traits. So I expect that we'll hear a lot about that exciting work today. As I just comment a little more on Hilary's background, she got her PhD at MIT in applied mathematics in 2017, and since then has quickly received a number of awards supporting her early career, including a Schmidt Fellowship at the Broad Institute and an NIH Director's Early Independence Award, which is really outstanding. One thing that is particularly intriguing to me about Hilary's background is that she did her undergraduate work in mathematics and then earned a master's degree in theoretical computer science at the Weizmann Institute of Science. And it was just 9 years ago or so that she was publishing papers like finite Voronoi decompositions of infinite vertex transitive graphs, which is excellent. So over the years, I found that biologists that have come from more theoretical mathematical backgrounds have really interesting perspectives on the problems in these areas. And I think Hilary certainly provides evidence for that association. So with that, I'll turn it over to Hilary, who will tell us a bit about, unless your title has changed, insights from complex trait fine mapping in diverse populations. So thanks for joining us today, Hilary.
Awesome. Thanks very much for that introduction and for this invitation. I'm really excited to be here. Yes, it was quite a shift from doing work of that sort to doing work of this sort. And I am extremely grateful to my PhD advisor, Alcas Price, for shepherding me through that transition. One logistical note, I would love to be interrupted with questions. I understand that this is a pretty heterogeneous group that's interested in a lot of different stuff. And so I'm hopeful that my talk will be accessible, but if at any point more clarification would be needed, then please interrupt me, even if it's just for that and not, other kinds of questions. So today I am going to talk about genome-wide association studies, which is the main focus of my research group. And let me start by talking about what a genome-wide association study is, and then I will pause and say why it is that we think GWAS is a really potentially transformative tool for understanding human disease. So in a genome-wide association study, we typically have cases and controls for a particular disease of interest, and then we genotype everybody using a standard SNP chip. And the result is data on order of magnitude, 10 to the 5 to 10 to the 7 common SNPs, 10 to the 4 to 10 to the 6 individuals. And then in a GWAS, you just go one at a time and you ask, is this SNP significantly associated with disease? Is this SNP significantly associated with disease? And you just get a p-value for every SNP one at a time. And because you're testing so many hypotheses, the standard for a significant association after multiple testing is a pretty rigorous 5 times 10 to the minus 8. And so we're looking for p-values that are smaller than that. So moving from cartoon to real data, here's an example of what the results of a genome-wide association study often look like these days. So here on the y-axis, we have significance of association. So this is the minus log 10 transform of the p-value. Higher is more significant. And the red line is 5 times 10 to the minus 8, that threshold for significance that I mentioned. And here, every point is a variant arranged. The x-axis is genomic coordinates with the chromosome marked. So now we have the entire genome visualized here along the x-axis, 1 point for every common SNP in our study that passes p less than 0.001, and then higher is more significant. And so what you can see is we, in this example, and for many other common diseases and traits, we're now finding tens or even hundreds of genome-wide significant loci. And just to emphasize how exciting that is, each one of these diamonds representing a peak, this is an area in the genome where we now have really robust, really reproducible evidence of an association between inherited genetic variation at that locus, at that region of the genome, and in this case, schizophrenia. Schizophrenia doesn't change your germline genetics. Most of the diseases and traits that we are interested in studying don't change your germline genetics. And so, and also the field has gotten very good at accounting for confounders such as ancestry. And what that means is that we can actually make really strong causal claims. There's something going on in these GWAS loci that causally contributes to disease risk. The effect sizes tend to be very small. And so, and there's many different loci. As you increase the sample size, we find more and more loci. And each one of these loci, though, contains a variant that causally contributes to disease risk. So this is very exciting. This has now become a study design that is very robust and reproducible and has identified tens of thousands of disease-associated loci for many different diseases and traits. And that brings us to our central problem, which is how exactly do genetic differences at these loci contribute to disease risk? We know, and we can be very confident now, that within these loci, if you inherit one haplotype instead of another haplotype, your risk for disease will be slightly higher. But why? What are the actual genetic variants that are causing this change in disease risk? And what are the genes that mediate that? And then how do the genes mediate that? What pathways, what networks, what is being disrupted that leads to disease risk? So I think of each one of these loci as a really exciting key, like a really exciting starting point that we can build something that's very reliable on. But we still have this enormous mystery. How do we go about building on this foundation? How do we really learn biological insights from these association data? So to talk about, so there's many different aspects to this problem. What I think of as a starting point is how do we identify the individual base pair level variants that are contributing to disease risk? And it was pointed out to me recently that a lot of folks who have been thinking a lot about COVID and not thinking a lot about GWAS think of variants now as like you can have like a variant, like the Delta variant or the Omicron variant or whatever. So here when I say variant, I mean like SNP or small insertion or small deletion, like the actual mutation, the individual mutation in the genome. Okay, so let's zoom in on one of these green diamonds to understand why it is that we need these variants. So if we look at a genome-wide associated locus, then it might look something like this. So now it's the same x-axis, the same y-axis. We've just zoomed in on one particular part of the genome. And you can see that, okay, this is, a p equals 10 to the minus 14. So a lot of things are highly significantly associated in this locus, but it's really complex. There's tons of stuff here. Like many of these variants are all significantly associated. And so how do we go about progressing from here? So what we think is going on is that typically there's only one or a small handful of variants that causally contributes to disease at a non-negligible level in a locus like this. And that it's only because of these one or small handful of effects that we're seeing a signal like this. And the reason that we see so many of these variants associated is because of linkage disequilibrium. So if this variant causally contributes to disease risk and it's inherited together with a bunch of these other variants, then all of them together are, each one of them is going to be associated with disease. And so it's really to be expected, even if there's something really simple going on, like a causal variant or two causal variants, we would still expect, just because of how GWAS is designed, that we'll see lots and lots of variants that are all significantly associated to disease because they all get inherited together. So the goal of statistical fine mapping is to try to recover the actual causal structure, to go beyond this association where we look at one SNP at a time and ask if it's correlated with our outcome of interest, and instead actually do the joint Bayesian modeling to try to uncover which variants we think are actually driving the association, to try to make claims about causal genetic variants. This is a problem that has been very well studied in lots of groups, and there's been some really cool methods development by other folks thinking about how to solve this. State-of-the-art methods are sophisticated Bayesian methods that typically have two outputs that we'll focus on. One of them is a posterior inclusion probability, or PIP. And so this PIP, which I'll refer to many times during the talk, is the probability of an individual variant to be causal according to the model. And then variants are grouped together into credible sets, and each credible set reflects one underlying causal variant. And the credible set is the uncertainty around which variant is that causal variant. So for example, in this panel, what we're seeing is that the algorithm, when it's given access to this data, it is very confident that the blue variant is causal. It's got a credible set of 1, and it's got a pip of 1. So the pip of 1 means the posterior probability that this blue variant is causal is 1 under the model. But then the algorithm says there's a second causal variant here. It's one of these 5 variants, probably, but we don't know which one it is. Like about 50-50 that it's this one, but we're not really sure. It might be this one, or this one, but we know there's only one other one. It's one of these 5. So that's what the credible set means. The credible set means it's one of these 5, not more than one, and the pip tells you how likely do you think it is to be each one of these variants. So I'm going to pause there for a second. On this background of why do we want to do GWAS? What does GWAS give us? And now why do we need to do fine mapping? And what does fine mapping give us? Are there questions so far?
So just a question. So from the fine, and this is so good so far. So basically from the Bayesian modeling, the credible set would just be those so why would they be 2 instead of 1? So that blue one is carrying most of the weight of the significance, right?
Yeah, one way to think about it, some intuition, an intuition that would speak to some folks is if you condition on the blue variant, there's still residual signal left. So you can be pretty confident that the blue variant is causal and still recognize that it's not sufficient on its own to explain all of the signal. Another way to think about it is if you think of Bayesian modeling as saying, what's the likelihood of the data given this variant is causal, or given these 2 variants are causal, or given that these 3 variants are causal, and searching through for the combination of variants that best explains the data. What the model is saying here is the blue variant together with any one of the red variants does a pretty good job of explaining the data, but they all do a pretty comparable job, and the blue variant by itself doesn't quite cut it. So the likelihood times the prior is higher if you allow for 2 causal variants here. Does that answer your question?
Okay, yes, including both red and blue all together, they explain much more than just the blue by itself. Okay, that's
exactly.
Thank you.
Can I quickly ask, you passingly mentioned that if there aren't germline mutations, that probably means these SNPs are causal to the disease. Can you explain that a little bit, like why that is, or if I misunderstood that statement completely, please correct me.
Sure. Yeah. So what GWAS tells you is folks that inherit a particular allele are tend to be people who have a higher rate of a certain disease. And so then we can ask, what's the causal structure there? And here are some possibilities. One is the genetic variant causes increased risk of disease. One is increased risk of disease causes the genetic variant. And the other is there's some confounder that causes both the genetic variant and the increased risk of disease. And in this case, we can rule out the disease causing you to have inherited a different variant. If you get the disease, that doesn't change which variant you inherited. And as far as confounders go, we actually have a pretty good understanding of ancestry as a confounder and how to correct for it pretty well. And when you're thinking about what kinds of things can confound the variants that you inherit, it's a pretty short list other than, well, okay, it's a list that consists of ancestry. And so I think that, if we were looking at gene expression, then that's a lot tougher because a lot of things can affect what genes get expressed. But not a lot of things can affect what alleles you inherit. And so if something is correlated with an allele that you inherited, as long as you've accounted for ancestry properly, then it must be that inheriting that allele affects your outcome in a causal way.
Got it. Thank you.
I should add one caveat around that. There's been some recent literature that's really cool about how it could be that if there's an allele that affects parental behavior, then you might be likely both to have inherited that allele and to have had a parent who had that behavior. And I think that that's something that I don't know to what extent that would affect these results. My guess is that's a pretty minor contributor, but I'm realizing I should add that caveat around the idea that their only possible confounder is ancestry. But yeah, so that would be another possible confounder.
So basically, sorry. So basically, then we can say causally for this, but like for SNPs, but for gene expression, for instance, we can't. But as long as we account for the ancestry, controlling for that, then we can.
Right.
Okay.
And I'm not talking about confounding by ancestry in this talk much, but that's something that the field, at least in biobank studies, at least in studies where one person has access to all of the data as opposed to a meta-analysis, it seems like we've got a pretty good handle on that. And one way to show that is by the high replication rates of GWAS findings.
I just have a question. When we have a complex rate, however, we should have several regions like this. So when we want to run a Bayesian model, we want to shrinkage some SNPs. Do you suggest to run all regions at the same time or each region separately? Because when we run the all region, we shrink all region toward the 0, but when we run single region, we just shrink some variance to 0.
Yeah, that's a really good question. So typically we run one region at a time because these methods are so computationally intensive that it's hard enough to run them one region at a time, let alone, feed in more of the genome all at once. We do want to incorporate information from the rest of the genome. And so sometimes we use mixed model summary statistics, which maybe I won't go into that. That might be, more detail than you're interested in, but there are ways that we kind of incorporate information from the rest of the genome indirectly into our models without actually jointly modeling the whole genome at one go with these fine mapping methods. But then, but the model assumes sparsity. So it's different from other standard Bayesian variable selection approaches because we really care about the posterior being calibrated. We don't want to just throw lasso at this and have it find some sparse solution that does a pretty good job. If there's 2 variants in perfect LD, we want the model to say, I don't know if it's this one or this one. I'd give it about 50-50 rather than just choosing one at random. And so I think that's part of why this problem is harder than a lot of other kind of standard sparse regression approaches that people think about outside of genetics. And that's why we need these really sophisticated algorithms that we can only run one mix at a time.
Mm-hmm. Thank you.
Great. Well, I will keep going. But again, feel free to stop me with questions. So why do we care about fine mapping? I've told you so far, GWAS identifies these regions of the genome that are associated with disease. Now we can apply these Bayesian fine mapping algorithms and zoom in on the variant level to find like actual base pair changes that we think are causally contributing to disease risk. Why bother with the second step? Why do we care about what the causal SNPs really are? Well, one reason is to enable, the main reason, I guess, that I'm interested in it is because it enables experimental and computational approaches to identify causal genes. And so if what we really want to do is come up with genes that we can do follow-up experiments on, or lists of genes from GWAS, then if we can find the fine mapped variants, if we can find the causal variants, then it now becomes a question of gene regulation, of linking SNPs to genes. And we've now got a handle that we can start to try to understand, okay, does this variant disrupt binding of a particular transcription factor? Does it disrupt, expression of a particular gene? And so it's a very useful tool for this important goal, but as I've mentioned, it requires very large sample size. It requires individual level data. It requires very heavy computation. And just to add some detail to the large sample size piece, GWAS these days have tens of thousands, hundreds of thousands, in some case over 1 million individuals, that's just to identify the associated loci. Actually resolving the signal down to base pair level resolution requires even more power. And so fine mapping, in order to get any confidently identified SNPs, you need either large sample size or a large effect size, or both. And so the need that we saw was for an atlas of fine mapping results. There have been really, a handful of high impact fine mapping papers where one person, or where one research group really undertook to put together data with large sample size that was high quality, and then do the computation and look at the results carefully. And what we wanted to do, was to understand the state of methods and then do a systematic scan at high sample size, many phenotypes, multiple populations, and be able to really put out an atlas, including a large compendium of results from fine mapping algorithms, so that we could provide a large foundation for people to start to build off an understanding from these two laws. So Masa Kanai and Jacob Euler are PhD students in my group. Masa is co-advised by Mark Daly and Jacob is co-advised by Freddie Savetti. And they set out to perform fine mapping in 3 biobanks: Biobank Japan, FinGen, and UK Biobank. And we're really excited about biobanks for fine mapping, because biobanks are an example where we have really large sample sizes shown here, and where we have access to individual level data. And we have that for many different phenotypes. And so we analyzed tens of phenotypes, and here you can see the histogram. It includes 26 phenotypes that were measured in all 3 of the biobanks, along with several other phenotypes that were available in only 1 or 2 of the biobanks. And so with this data in hand, we applied state-of-the-art methods for first doing large-scale simulations to benchmark these methods in as realistic a setup as we possibly could put together. And then once we had convinced ourselves, that these methods were good ones that we wanted to use, then with about 1 million compute hours, we fine mapped over 15,000 genome-wide significant loci for all of these diseases. And what we got was 30,000 credible sets. And so remember, each credible set harbors a causal variant according to the algorithm. And within the credible set, we can say how confident the fine mapping was by asking what's the best PIP in the credible set. So for example, breaking down these 30,000 credible sets, you can see that nearly 5,000 of them harbor a variant that had a PIP of 90% or greater. So that means that as a result of the analysis that we did, we found over 5,000 cases where we could resolve down to a single variant with 90% confidence that that's a causal variant in the locus. So this is really exciting, right? This is now something where we've been able to take advantage, build on top of a lot of the progress that the field has made in recent years, and then just brute force apply it to a ton of data, and now get this really large resource where instead of talking about loci, we can now talk about individual variants. So we wanted to get a sense for, to validate these results in some orthogonal way. And so one way to do that is to take advantage of the fact that the fine mapping algorithms don't know about functional annotations. So we never tell the fine mapping algorithms which variants are coding, which variants are regulatory, which variants have no known annotation. But if you break up the genome into these different categories, CRE is cis-regulatory element, and then you ask what proportion of the lowest confidence variance falls into each category. So these are variants where the algorithm thinks there's less than 1% chance that the SNP is causal. And then you bin all the way up to the set of variants that the algorithm thinks is more than 90% likely to be causal. Then you can see that even though we never told the algorithm which variants were coding, it's finding a really big enrichment for coding variants here in each of the subsequent, in each of the bins. So this is pretty exciting validation. These algorithms are really giving us something that has a lot of signal and that's able to point us in the direction of mechanism, even though all it has access to is the genotype and phenotype information. Before I go on, maybe I'll just pause there one more time for questions. So now we've got this large set of fine mapping results, this large set of individual variants with confidence at varying levels for being causal for particular phenotypes in these 3 different biobanks. So now that we've got this, what can we do with it? One thing that we can do is ask about variants that affect more than one phenotype. So what this plot is showing is, now again, we have the whole genome arrayed from chromosome 1 up to chromosome 22. This is an analysis of autosomes. And for each SNP, we now have the number of traits in which it was fine mapped. And we've grouped those by category. Some traits are highly correlated with each other. So if something gets fine mapped for both HDL and LDL, you might not be that excited. But if you get something that's fine mapped for a metabolic trait and a lipid trait and a hematopoietic trait, then that starts to become more interesting. And so you can see GCKR, for example, harbors a variant, which is a missense variant that's annotated here. A conserved missense variant in GCKR was fine mapped for, I don't know, is that 27 traits or something like that, including traits in the metabolic, lipid, cardiovascular, hematopoietic, hepatic, and renal, and other categories. And this is concordant with what a lot of other human genetic studies have shown. There really is widespread pleiotropy, a lot of genetic variation that contributes to phenotypic variation in many different domains. And several of these genes are very well studied as well. SLC39A, ApoE, there's a lot of familiar gene names here that are known. It's understood how it is that they contribute across all of these different categories, but now being able to do something systematic and large scale and then putting out all of these results lets us kind of zoom out and take a peek at the top genes that in our analysis are showing the most pleiotropy. And if we then ask just for a summary of this data, so how many domains are the different SNPs fine mapped, you can see that this is now a log scale y-axis, but there's over 1,000 SNPs that are fine mapped in at least 2 domains, and then SNPs that are fine mapped in up to 7. One reason that this pleiotropy, as it's called, is so exciting is because it gives us a starting point to try to learn about other diseases from our GWAS results, from our fine mapping results. So we fine mapped traits in these 3 biobanks. None of these biobanks were ascertained for any particular disease. So we have excellent data on biomarkers, anthropometric traits, and very common diseases, but we're not well powered to study autoimmune or psychiatric diseases that are less common in the population. But folks who are particularly invested in those diseases have gone through enormous effort to put together very well-powered meta-analysis studies, many of which have been cataloged in a website called Open Targets. So Open Targets now has this really, treasure trove of data on very well-powered GWAS for many different common diseases. Over 1,000 phenotypes is what we looked at. And what it gives us is for each locus, the index variant, the most significant variant, and then all variants within R² of 0.7. And the problem is the data that's available for these studies is not enough to do fine mapping on. It's well understood now that there's often more than one causal variant in a locus. It's also well understood that if you want to do really reliable, high quality, multiple causal variant fine mapping, you need in-sample LD, and that's just not available for these data. But we can ask if our biobank fine mapping can be useful. And in particular, we can ask if we say that we think it's pretty likely that either the index variant or one of the variants in R² greater than 0.7 could be plausibly causal, we just don't know how to differentiate among them. Well, if it turns out, for example, that you've got a highly significant variant for COPD that in our analysis is fine mapped for a measure of lung function, like FEV1 over FVC, then you might be hypothesized that that's the causal variant. These signals are actually reflecting the same underlying causal variant, and by analyzing the biobanks, we've been able to identify which variant that really is. And so that's what we did, is look for fine mapped variants in the biobanks that are highly significantly associated according to the meta-analyses and Open Targets. And here's the results of that analysis. You can see that the darkest is up to pip equals 0.9, and then the lightest is pip 10% or greater. And you can see that for a lot of these phenotypes, we're able to find many significantly associated loci for which we can nominate a possibly causal variant on the basis of our own fine mapping within these biobank traits. Maybe I'll pause there one more time before I go on to talk about cross-biobank analyses.
Some of the variants that you looked at when you looked at their functional annotation, I'm assuming CRE was like cis-regulatory elements. It sounds like you're focusing on looking at these like fine map variants at like genic regions. Have you looked more at like maybe enhancers or super enhancer regions? Those are the CREs that might, those I imagine probably affect multiple genes, and maybe they're also like multi-domain or pleiotropic. I think that was the word you used.
Yeah. That's a great question. So the way that we are defining cis-regulatory elements is open chromatin and H3K27 acetyl. So Jacob assembled a bunch of open chromatin data sets from a lot of different sources and a lot of different cell types and tissues, and then overlapped them with H3K27 acetyl. I think it was from Roadmap Epigenomics, Union, again, over as many cell types as we could find. And so those tend to be closer to genes than usual, but that does include some more distal enhancers as well. We don't see as strong enrichment in aggregate over cell types for that, but in other work that I've done previously, we found really very strong enrichment if you zoom in on the enhancers that are active in the cell types that are specific to the phenotypes. And so I think that's definitely like a really important thing, like way to be characterizing these results. And in a later slide, I'll get back to the regulatory variants and how, some of the ways that we've tried to analyze those and decompose them a little bit more. I see, but one point that you're making is if you look specifically at the pleiotropic variants, then we might be likely to see more enhancer variants because they can affect multiple genes, which might then affect multiple traits. And I think that's an interesting hypothesis. We find really a lot of coding variants. You can see here, this again is analysis that doesn't know about annotation, but it's still finding of these top variants here that like half of them are missense variants or something. So it looks like we're still finding a lot of coding stuff, but I think understanding how exactly the non-coding variants are acting is, a really important question.
Cool, thank you.
So one of the reasons that we were really excited to have done this fine mapping in 3 separate biobanks was that it allowed us to try to replicate our results. To the best of my knowledge, no one before has had enough data that they could do independent fine mapping in 2 separate cohorts and then be well enough powered that you could actually expect to find results in both of the cohorts, that you could use one to replicate the other. But we were able to do that. And so we looked for variants that had a pip greater than 0.9 in one biobank, and then also had a pip greater than 0.1 in at least one of the other biobanks. And these replicated variants represent our highest confidence set. These are the variants that we can really say with, it's not just one data set, that we're really identifying these variants in a robust way. And here's the breakdown. And continuing with this pattern, we're finding coding variants are again really overrepresented in this set by a good bit. And then just to pull out a couple of examples from this exciting set of variants that represents our highest confidence results. It includes well-known variants like coding variants in ApoE, coding variants in NOD2 and in MARC1, a 3' UTR variant in TP53 for skin cancer. One interesting story that Masa pulled out was a synonymous variant in the gene ASGR1 that had pip greater than 0.9 for a liver biomarker, alkaline phosphatase, in both Biobank Japan and UK Biobank. And then looking into another data set, GTEx, looking at RNA-seq data, we found that this variant was actually a splice QTL for ASGR1. And then Masa found this paper from 2016 in which a nearby non-coding deletion that was also associated to ALK-Phos as well as other phenotypes, including coronary artery disease, had also been shown to affect splicing of ASGR1. Moreover, this is an independent variant. If I remember right, it was monomorphic in BBJ. I forget exactly what the line of evidence was, but these 2 variants are definitely independent from each other. And so this is an exciting example where we've got 2 independent variants that are both close to the same gene that then converge on the same proximal mechanism, which is disrupted splicing of ASGR1. And so that kind of sets the stage for now trying to understand how it is that disrupted splicing of ASGR1 is leading to these changes in liver biomarkers. But it gives us, we've gone from associated locus to confidently identify the variant to proximal mechanism. Being able to take each one of those steps is really hard. And so being able to find two variants converging on the same proximal mechanism is something that we're really excited about. One thing that you may notice is that this isn't actually a lot of variants compared to the large set of variants that we started with. We weren't able to replicate nearly as many variants as we thought we were going to be able to. And there are several different contributing forces to this. One is it turns out that requiring replication at pip greater than 10% is pretty stringent. A lot of these, they're associated, but they're not associated at a high enough significance level that you would expect to be able to replicate the fine mapping to begin with. Another contributor does seem to be that the fine mapping algorithms have some way to go in terms of reliability. And maybe when the algorithm says 90% posterior probability of being causal, what that actually means is something high, but not quite 90%. So we do think that the algorithms aren't perfectly calibrated in the pips that they're reporting. But one of the main contributors was that a lot of the variants that are fine mapped in one population are rare or monomorphic in the other two populations. And so one way to think about this is if you have a variant and it causes a phenotype, then selection might be pushing it down in allele frequency to make it harder to find, but there's still some randomness in what allele frequency it has. And if you only look at one population, you just get one sample from that distribution. But if you look at several different populations, then that variant now has more chances to rise to a higher allele frequency. And if it rises to a higher allele frequency, you're better powered to detect and fine map it. Moreover, both Finland and Japan are bottlenecked populations, which means that there was a point at which a deleterious variant where selection is pushing the allele frequency down had an opportunity to suddenly jump up in allele frequency due to the bottleneck and subsequent rapid population growth. And so we found that indeed our high pip variants were disproportionately these bottlenecked variants. And I think that this is a really cool finding. So let me walk you through the figure really carefully. So first, let me tell you what allele frequency enrichment is. So allele frequency enrichment is just a ratio of allele frequencies. It has nothing to do with a phenotype. It's just saying for a particular SNP, what's its allele frequency in Finland divided by its allele frequency in our best approximation to the ancestral population before the bottleneck, in this case, non-Finnish, non-Swedish, non-Estonian Europeans, NFSE. And then you add 1 so that you're never divided by 0. And what that means is that the variants off to the right are the variants that are much more common in Finland than in this NFSE ancestral population. And similarly, this is the allele frequency enrichment, the ratio of allele frequencies in Japan to non-Japanese, non-Korean, East Asian populations. And this is using data from NOMAD. And so using this data, one thing to notice is just if you look at all of the coding variants, and again, this still had nothing to do, I haven't introduced any phenotypes or fine mapping yet in figure A. If you just look at the coding variants, there's this right shoulder. There's a bunch of coding variants that have an allele frequency enrichment where they've got higher allele frequency in Finland than in the ancestral population, and slightly less pronounced, but it's definitely also there in Japan. We would expect the set of variants to be enriched for deleterious variants. And sure enough, if now you introduce the fine mapping results and you ask, what does this breakdown look like for our variants that are the least likely to be causal all the way up to the variants that are most likely to be causal, you can see that in our top most likely to be causal variants, we really have an enrichment for these bottlenecked variants off on the right. So, 36% of the variants are passing this 10-fold enrichment, as opposed to just 21% that you would expect by chance. And so this reflects the idea that there were a lot of variants that were in an ancestral population causing disease, got pushed down to a low allele frequency, went through the bottleneck and jumped up in allele frequency so that now they're much higher in allele frequency in Finland than in the ancestral population. But then haven't had enough time yet for selection to successfully push them back down again. And so that's why we're now able to access those variants. Like those variants now, they're rare or monomorphic in the ancestral population, but they're common in Finland. And so now when we're fine mapping for a particular disease, we're powered to detect these variants. So some example of these bottlenecked variants include a Japanese enriched missense variant on CD36, which we identified as fine mapped for platelet count, and which is a known pathogenic variant for CD36 deficiency, which has higher prevalence in Japan, and which we identify in our study. Similarly, there are 2 novel coding variants in a gene called PLOD2 for height that were enriched in Japan and in Finland, respectively. So this kind of shows the power of analyzing data from these bottlenecked populations for identifying variants that in many other populations are rare because they're deleterious. This is a benefit. So it hurts us on the one hand if we're trying to replicate our results, because if you fine map a population enriched variant in one population, then it's not common enough. You can't replicate it in the other populations. On the other hand, it gives us access to many more variants that we wouldn't have access to otherwise. And often they converge on the same gene. And so here's an example of that. This is the gene ABCG2, which is a known gene for gout. Here's the gene with the exons and the introns. And then each of these gray diamonds is a variant that's been fine mapped. And then the color shows which cohort it was fine mapped in, and then the y-axis is giving the PIP, so the confidence with which it was fine mapped in that cohort. And so here you can see there are 2 missense variants that were fine mapped at pretty high confidence in UK Biobank, but not in the other 2 populations. Turns out they're rare or monomorphic in the other 2 populations. Then we've got another missense variant, which is common in BBJ, but rare or monomorphic in the other 2 populations. One that's common in all 3 populations and is fine mapped in all 3 populations, just in UK Biobank, it's only at 5% confidence. And then a stop gain variant that again is common in only one of the biobanks and rare in the other 2 biobanks. And so this shows that by putting together data from these different biobanks, we now have access to the different variation in the different biobanks. But they're all converging in this case on one gene, giving us this amazing allelic series of 5 separate missense variants, all on the same gene, all identified in different combinations of populations. And this isn't the only example of that. We found these types of allelic series in many different genes. So we found here's the number of predicted loss of function or missense variants that were identified, and each of these genes colored by which cohort they were identified in. And you can see that combining data across cohorts really enables us to put together much longer allelic series than what would be possible otherwise. In total, we identified over 1,000 unique fine mapped coding variants in 969 genes. I'll pause there again and ask if there's any questions on this most recent part.
So potentially, if you were to get data in a brand new population and you found a bunch of variants, and if it's near some of these variants that cluster together, do you think it would be reasonable to be able to say like it has a similar functional consequence?
Yeah, I think that's super interesting. I think there are some cases where that's an easy statement to make, like I don't know, it's coding in the same gene, so it probably affects the same gene. I think that if you have a non-coding variant that is pretty close by, then it kind of turns into intuition. Like my guess is like, yeah, probably most of the time, if they're pretty close, then they're doing, related things, but I don't have any good evidence or a way to support that particular claim.
Got it. Cool. Thank you.
Just a clarification question on the definition of monomorphic. Is that a SNP that's specific to that one population, or?
Yeah. So monomorphic as opposed to polymorphic, like it doesn't vary. There is no variation. Allele frequency is 0.
I see. So of all those individuals sampled from that population, they all had that.
They all had the same allele. Exactly.
Thanks.
And so if everybody has the same allele, you can't associate it with anything or fine map it. Yeah. So we were very excited about these coding variants. You know, interpretation of GWAS is really difficult, but when we get a coding variant, or better yet, an allelic series, it makes our life a lot better because it just points us directly to a gene. But much more often, we get non-coding variants. And so then what do we do? One common hypothesis is that these variants act by regulating the expression level of genes nearby. So depending on which allele you have, it's not that you change the protein coding sequence of the gene, but you just get more or less of it. And so there's a data set called GTEx that has RNA-seq data in 49 tissues. And we applied the same fine mapping algorithm that I just described to GTEx in order to identify individual variants and credible sets that are causal for changes in gene expression. So this is now totally separate from all of the complex trait disease biobanks. We're only asking now about gene expression as a phenotype. And so each one of these variants in the red, for example, is a variant that's got a pip greater than 0.9 for the phenotype expression level of gene X. Once we've got all of these variants that are expression modifying variants in a tissue according to GTEx, then we can integrate these results, and we can ask how many of the variants that we're identifying in our biobank fine mapping are we also identifying in our EQTL fine mapping? And in particular, using a rather conservative approach of multiplying the pips together, which actually you can derive starting from some conservative priors using a Bayesian analysis, you can identify co-localized variants. So these are now variants that we think are causal for a particular disease or complex trait, and also causal for the expression level of a particular gene. And then if you focus on the variant, we'll call that a co-localized variant. If you ask, well, which gene is that? Then we'll call that the co-localized gene. And a common way to interpret these types of results is if you've got a variant that causally affects a disease, and then you know that it causally affects a gene, then we typically figure, well, probably that gene is mediating the disease. How much could one variant do? It must be that the way that it affects disease is via this gene that it also affects. And that turns out to be oversimplistic, as I will say in a couple of slides, but that's one of the big motivations for why a lot of folks are interested in this type of EQTL complex trait co-localization analysis. That was a little bit complicated, so I'm going to pause again and see if that was clear so far. Why would we be interested in fine mapping gene expression?
So just a question. So you're saying that like the results from the GWAS EQTL co-localization analyses could be oversimplistic as well, like kind of believing that the association between the SNP and the gene expression and the SNP and the disease to link the SNP disease and gene together, that might be too simplistic. Is that right?
Yeah, exactly. And I'll show a slide shortly that points out that a lot of these variants do more than one thing. For example, they often regulate more than one gene. And so if you happen to only identify one of those genes, like you just by chance, or if you analyze the wrong tissue or something, then it could actually be pointing you in the wrong direction, because a variant can do a lot of things, and it's hard to say which of the things that it's doing is the one that's leading to the disease. I'm running a little bit low on time. Actually, I think I have enough of this. Okay, so now we could ask, looking at these variants that are fine mapped both in UK Biobank and for GTEx, and this analysis was done at a time that we only had access to UK Biobank, so now it's only a single biobank. This now is the enrichment of how much more common was it to have this annotation, given that you were a co-localized variant or a high PIP variant in GTEx or UK Biobank. And you can see that for loss of function variants, we have really high enrichments for these co-localized variants, as well as for UK Biobank. And there's still enrichment, it's just a little bit lower for GTEx, as you might expect, because loss of function variation, that's not like the most common thing you would expect to affect expression levels. And similarly, for missense variants aren't much more likely to be expression modifying, but they do contribute a lot to our complex traits. And then for these regulatory variants, this is where we start to see a higher enrichment for the expression modifying variants and for the co-localized variants than we do for the complex traits. And one thing that we were interested in was also this final non-annotated category where we see that there's actually a smaller proportion of these co-localized variants that are non-annotated than in either of the original two subsets. And so that means that once we zoom in, particularly on the variants that are fine mapped for both GTEx and UK Biobank, many more of them are falling into one of these categories. And so it could be that this is a particularly high confidence set of variants again. The variants, the co-localized variants in general tend to be very common. This is because GTEx has pretty small sample size. And so we're looking at minor allele frequencies that are usually above 5%. And this is in contrast to the variants that we're fine mapping for complex, the coding variants that we're fine mapping, which many of which have minor allele frequencies less than 5%. And this leads to similar rates of co-localization in the 3 biobanks, even though our EQTL data has a European bias. This is kind of just a side note about co-localization for folks who are thinking about how it behaves in different ancestral populations. One thing that we wanted to ask was, okay, so now I've told you we fine mapped complex traits. We identified a bunch of variants that we believe are causal for disease in complex traits. Then we fine mapped GTEx and we've narrowed in on a subset of variants, co-localized variants that we think are causal for complex traits and causally modify expression levels of particular genes that we can name. And now we can ask for this specific set of variants, how are they doing this? Are they disrupting particular transcription factor binding motifs? Are they changing patterns of open chromatin? Is there some way to get at the most proximal? What's the first effect that this variant has? What's the what function is it having that's causing it to have these downstream consequences? There's a lot of data here that we tried to incorporate. So for example, does it disrupt a transcription factor motif using a database called Hokomoko? Is it in a ChIP-seq peak for the transcription factor? So we can try to further restrict not just to motifs, but to motifs where there's additionally evidence that the TF is occupying that particular stretch of base pairs. We looked at this new method called Informer, which is a neural net-based predictor of which variants are likely to disrupt transcription factor binding or accessible chromatin. And then we used a type of data called allele-specific binding, which I won't go into for now, as well as a new database of DHS footprints. So this is looking just at patterns of open chromatin for folks who are familiar. If you've got chromatin that's exposed and then a little footprint where it's not exposed and then more where it is exposed, then that often reflects the binding of a particular transcription factor leading to that footprint. And so then we could ask for the green is the co-localized variants, the blue are the complex trait variants in general, not restricted to co-localized variants, and the orange is the control, the least likely to be causal. What proportion are we able to annotate with each of these different sources of data? One thing that we found was a lot of the standard ways that people would think to do this, for example, just looking up transcription factor motifs in a large database like Hokomoko, have a very large background. There's more than half of the variants in our control set were predicted to disrupt TF binding if you don't put any other filters on it. If you look here, though, where you're now requiring it not just to disrupt the motif, but also to have ChIP-seq evidence that the transcription factor actually binds at the motif, then you get a much lower background, a nicer enrichment, but you're not able to annotate as many variants. So we looked also at this accessible chromatin footprinting, at allele-specific binding, and at accessible chromatin, allele-specific accessible chromatin. And we found moderate enrichments, varying levels of background. I think the main takeaway, the thing that surprised me the most, other than this very high background for transcription factor motifs, was that Informer, this neural net-based method for predicting the effects of variants, actually did really well with a pretty low background and high enrichments. And by just combining methods requiring, for example, that 2 or more methods support a proximal function for this variant, we were again able to recover low background rates and then annotate a pretty large fraction of all of these variants. So I think that this is cool because a lot of times when you're trying to figure out what a variant does, it's kind of hard to there's lots of different ways that you can try to do it. You can try to do it with EQTLs, you can try to do it with TF motifs, or you can, try out Informer because it's new. But being able to do a systematic scan where we first say, let's take all of the complex traits, and then let's take all of GTEx, let's identify a large set of co-localized variants, and now take many of these different data sets that can be used to characterize the variants and just systematically say, what is the background level? What is the enrichment? How many of our variants are we able to annotate as predicted disrupting transcription factor binding? And then to wind up with something where you've got a pretty low background and a pretty high rate of annotation, I think is pretty exciting. So that's characterizing the co-localized variants themselves. Another thing that we might be particularly interested in, as we've discussed, is the genes, so we have these variants that affect the transcription levels of particular genes. So what can we say about those genes? One reason, this is what I was just saying earlier, it's hard to say if you've got a SNP that affects a disease and also the same SNP also affects a gene, well, can you say the gene affects the disease? And one reason that that's difficult is that SNPs often regulate multiple genes. For example, there's a non-coding variant independent of the coding signal near ApoE that was fine mapped for LDL and ApoB, and then is also fine mapped as expression modifying for 3 different genes in the locus. Another example is this SNP that's fine mapped for mean corpuscular volume and is also fine mapped as affecting the expression of 2 different genes in the locus. It's in the 3' UTR of one of them and in the last intron of the other. And so for these kinds of examples, we have no idea what's going on. I guess by we, I mean, I don't know, what's going on. Is it that there's some kind of coordinated program that it's because it affects these genes and these genes work together in some way that it's affecting the phenotype? Or is it that it happens to affect 2 genes and one of them happens to be really important and we need some other source of data to tell us which of the genes is the important one? I think this is a really important area to be thinking about. And then adding to the danger of interpreting these kinds of co-localization results, the direction of effect can be tissue specific. So even if you've got one trait and one gene, if you've got that trait gene pair in more than one tissue, then a non-negligible 6% of the time in our data, the direction of effect is not unanimous across tissues. So to give one example of that, there's a gene that we know causes liver disease in mice. And we've got liver biomarkers in our data set. And sure enough, we've got a high confidence co-localization between this gene and this phenotype in lung and in pancreas. But depending on whether you look at lung or pancreas, you can either draw the conclusion that decreased levels of this gene increase the biomarker on average, or that increased levels increase the biomarker. And that depends entirely on which tissue you look in. So this stuff gets very complicated very quickly, and I think it just calls for a lot of caution and thoughtfulness about how we go about integrating and interpreting results from data like this. So to conclude, in this project, we put together an atlas of likely causal variants. We think this is going to be a resource for helping to interpret GWAS. We were able to replicate some of these variants across populations in a way that increases our confidence in a new and unique way. By combining results across populations, we found a allelic series of coding variants in the same genes. We then integrated with EQTL data to identify fine mapped regulatory variants. And then we threw a bunch of different data at it to characterize what the proximal variant, what the proximal functions were for these regulatory variants. And we shed a little bit of light, adding to the already extensive literature on why it is that gene prioritization from GWAS is so complicated. So with that, I'd like to thank Masa and Jacob, who led the project, Mark, co-supervised it, our many collaborators, the funding sources, the participants of the biobanks, and you for your attention. I'm happy to take any final questions in the last, minute, I guess.
Thank you so much, Doctor. This was such a good presentation. I had a question. So if you have a set of SNPs and then those could have some regulatory effects, so do you recommend, I know that the Stanford has a great tool, but do you recommend any other tools for maybe perhaps like your atlas for doing enrichment analysis over a set of SNPs that could have regulatory effects?
You're saying, are there any data sets of SNPs for doing enrichment analyses, or
Yeah, that is for SNP enrichment analyses. Yes.
yeah, sure. So our fine mapping results for UP Biobank are public, and for GTEx, Biobank Japan, and FinGen are coming very soon. So this particular set of co-localized variants, for example, soon that'll be something that you can just download and look at if you're interested.
Oh, so that will be available soon, right?
Yes.
For that. Okay. Thank you.
Great. Thank you, Hilary. Let's see if there are some other questions. Let's see. Mark, you want to go ahead?
I think Malay had her hand up first if she wants to go first.
Oh, okay, yes, Malay, go ahead.
Oh, sorry. Hi there. Thank you for your talk today. Really interesting stuff. And I may have lost this, or may have not caught this, but how do you rectify for mitochondrial DNA?
So we don't, in this project, analyze mitochondrial DNA. So we're looking at SNP chips where we already know ahead of time that what we're looking at is autosomal genetic variation. So we haven't been analyzing mitochondrial DNA.
Okay, thank you.
Thanks for a great talk, Hilary. You mentioned that the PIPs may not be that well calibrated. And so I'm wondering, is it possible to do that? Can you start from a gold standard of known causal variance and basically create a calibration curve so you can get good calibrated or well calibrated PIPs?
Yeah, we've thought about this a good bit, and it's just really, it's hard for several reasons. One is it's hard to find a large, unbiased gold standard set of causal variants from GWAS. And then on top of that, the calibration probably depends a lot on sample size and effect size. Well, we have another project ongoing to try to understand why it's not calibrated and develop a method that's more calibrated. And while I love the idea of empirical calibration, I think just the availability of gold standards makes that pretty challenging.
Okay, Erica, did you have a question?
Yes. Thanks for talking. Thanks for answering questions along the way. For the gene prioritization problem, have you considered incorporating like chromatin interaction data or long range interaction data to basically say like, this is a variant locio that we're interested in, and see if that helps narrow down which genes might be ultimately linked to the actual complex trait?
Yeah, absolutely. So this is something that we've done a good bit of work on, and other groups have developed methods. So I'm thinking in particular, Jesse Ingrates at Stanford had a recent Nature paper on his activity by contact method for connecting enhancers to their target genes using Hi-C data, as well as enhancer activity data. And he has shown good results for integrating that with GWAS data for IBD. There's also been, a number of people have tried a number of different approaches for using different types of epigenetic data for this. For example, correlating enhancer and promoter activity across many different samples to connect enhancers to promoters. And so we recently took a bunch of epigenetic based methods and a couple of EQTL based methods, and then another family of methods where you try to look at properties of the gene itself and do systematic benchmarking of how well these approaches are doing across a large number of phenotypes. And it seems like no individual method on its own allows you to draw really high confidence conclusions. But that by combining many different sources of data, it's possible, you know, if the gene has an enhancer promoter connection, for example, from Hi-C data, and also it's expressed in a tissue that shows genome-wide enrichment for the trait, then you can start to be more and more confident in your conclusions. The relevant figures, figure 4 of weeks. So that's the analysis that I'm referring to.
Thank you.
Great. Thanks again, Hilary, for such a great talk. I had a question of my own. I thought I'd sneak in here. So you mentioned about how many of the variants were not replicable across the different biobanks, and you mentioned maybe a few reasons for that. I was wondering how much of that is expected just statistically, you know, I think you did some simulations, you know, how if you simulated all those biobanks again, how many would be replicable across those? Did you do that type of
We
experiment?
did that, yes. And it turns out that you still get pretty low levels of replication, but higher than what we're seeing. So we think that a lot of it is due to just statistical noise, but not all of it, unfortunately.
Okay. And what is the number? Maybe I missed that. Of what percent are that you can replicate?
Let me pull up the figure. I included it here somewhere. So if you take all variants that had high PIP in one of these three populations, and then first you ask, is it GWAS significant or not? Because if it didn't pass significance, then you're not going to find map it and you can't replicate it. And then only for the significant ones you ask, did it have PIP greater than 0.1 or not? Then across all of these different pairs, this is broken down by which population was the discovery and which was the secondary. But overall, we were only able to replicate 55%. And then if you ask, well, what about the ones that were not genome-wide significant? Why weren't they genome-wide significant? Well, a lot of them did have what we would consider to be a replicated association, just at a lower significance level of P less than 0.01. So that's the orange category here. So all of these, super plausible that they're causal, and it's just statistical noise that's leading us to not even try to find them. And then the gray parts are, basically these bottlenecked variants, all of the variants that we wouldn't be able to identify to begin with. And then to get to your question, here's the simulation results. So if you do simulations where you have all of the same, like all of these PIP greater than 0.9 variants, you simulate them to be causal, and then you try to map them in the secondary population, then the amount of fine mapping replication that you're able to achieve is not 100%, but it is higher than what we're seeing in the real data.
That's really interesting. So you think it's mostly due to the fine mapping algorithms, or is there another factor you?
Yeah, I guess I think that the difference between the blue and the green here, like the difference between the real data and the simulations, it is possible that the simulations are unrealistic in some way. Maybe there's a winner's curse problem, for example, where we're simulating larger effect sizes than what's truly in the real data. But we have some other lines of evidence that every once in a while, these fine mapping algorithms give false positives. And so I think that this is kind of concordant with some of these other results. And I just remembered we have this figure as well. So if you look at the variants that replicate and the variants that don't replicate, then this is now functional enrichment. So many fewer of them are annotated functionally. So if we consider functional enrichment to be one sort of orthogonal way to ask how real are these results, the ones that replicate sure seem to be a lot more real than the ones that don't replicate.
Great, thanks. That's a really great answer.
I think you're thinking about this exactly the way that we did.
Great. We're a little bit over time, but maybe is there any one last question? Okay, well, if not, let's all thank Hilary again for a fantastic talk. And thanks again for joining us, Hilary.
Thanks very much for your attention and invitation.